Hi everyone, my name is Iris Nava and I am a third year PhD candidate in the Molecular and Cellular Biology program at Brandeis University. I'm so excited to tell you a bit about my journey here as well as my involvement in research. Let's start with an overview of my life. I grew up in Southern California, relatively close to Disneyland, which was so much fun as a kid. The summer before I started high school, my family moved to the fabulous Las Vegas, Nevada. The picture on the left is Red Rock Canyon, a conserved desert area for hiking and rock climbing. And my high school was actually about 10 minutes away from Red Rock. This is Emma and Chloe. They're waiting for me to get back for winter break. After graduating from high school, I attended the University of Nevada, Las Vegas, or UNLV for my undergrad. And that brings me to now, attending Brandeis University for my graduate degree. I first realized my enjoyment for biology in middle school during my teacher's lessons of mitosis. Mitosis is the process by which a cell divides and produces two identical daughter cells. Now, this isn't exactly what got me excited about biology, but rather the fact that our cells are doing this constantly. This is all without us having to think about, and we can't exert control over it. I began to wonder what other processes occur in our cells without us knowing, and generally in the world around me. I continued to take as many science classes as I could because I was just so curious. I really had no idea that my interest in biology would lead me to where I am. Eventually, I started at UNLV as a biochemistry major, which is the study of the chemistry of life. It explores the chemical processes and substances in living organism. The field bridges biology and chemistry to explain how life works at the molecular level. This is an interesting field, but about 2 years in, I was having some difficulty with the material and just difficulty getting genuinely interested in the material. After talking with some friends and an academic advisor, I looked through the degree tracks for biochemistry versus biology and the various concentrations offered. The classes being offered for cellular and molecular biology just sounded so much more interesting and exciting to me. So I switched majors to biology with a concentration in cellular and molecular biology. Now, when I think of these two fields, I think of cellular and molecular biology as sort of zooming out a little bit, where I'm looking at overall processes and cells as a whole, not the exact chemical reaction that occurs to make it all happen. And I still got a minor in chemistry. A professor in one of my introductory biology courses always included areas of biology that scientists were not completely sure about, meaning these areas were active areas of research. And at the time, this blew my mind. I was on a quest to learn everything I could about biology because I loved it. I had no idea that we didn't know everything. So this only fueled my interest to keep going. Then a close friend of mine got involved in a chemistry lab doing research, which I also had no idea I could do. So I looked into a biology lab and I reached out to the same lab. And through the encouragement and help of Dr. Singh, I was able to secure multiple scholarships and fellowships to conduct research. The NSF-REU, or the National Science Foundation Research Experience for undergraduates in the top right, was the first fellowship I earned before I even knew that I liked research. It ran through the summer and was instrumental in getting me started in research. From what I know now, this scholarship or fellowship is still running and it's across the US in various schools. It's a great opportunity to get started in research and meet other professors as well as other young scientists. During this initial exposure to research, I learned how much time and how difficult research can be. But I also felt the excitement of learning an entirely new fact, and the excitement of being at the edge of what we know. Dr. Singh told me about the world of research and how I could actually make a career out of it. During this time, these scholarships and fellowships were how I actually got paid to do research. Each one ranged about $4,000 to $6,000 over the summer or sometimes during the semester. In the same lab, we were interested in eye regeneration, or they still are interested in eye regeneration in tadpoles. So I learned how to take care of the frogs and tadpoles. Now, I have some videos here of the African clawed frog, or Xenopus laevis. That is what they normally look like. And then I also have an albino frog. Here I have some tadpoles swimming in a petri dish. Now, I knew I wanted to continue doing research and wanted to find a way to make a career out of it. I knew I wanted to attend graduate school and earn my PhD, but for a variety of reasons, I chose to do a gap year. For me personally, felt I felt it was too much to do research, classes, and apply to graduate school all in my last year. I also felt I could use more research experience since a good chunk of my experience was interrupted by COVID. And I knew I needed to make it a productive gap year. So I talked to Dr. Singh about continuing in the lab, and with the help of two other wonderful professors, I was encouraged to apply to another NSF fellowship. I thankfully did earn the NSF research experience for post-baccalaureate students. A post-baccalaureate is a course of study or academic work after earning a bachelor's degree. For me, this fellowship looked like continuing research under Dr. Singh and being involved in helping and attending some of the RU sessions during the summer. There are a lot of post-baccalaureate programs at various universities that often look different. I got paid for my post-bac. I'm not sure that's the case for all of them, but know that there are paid post-bac programs out there. During my time as an undergraduate and as a postbac in Dr. Singh's lab, I got to present my research at various poster sessions at UNLV, both in person and virtually at Harvard, and at a large conference called AberCamps. I did have quite some struggles to get to where I am now, and I still deal with these things today. When I was applying for grad school, I was worried that maybe I'm not smart enough for grad school, or what if I can't get into a grad program? My mom sat me down and told me, you just got to throw the seeds and see what grows. These, and that really helped me get through and apply to graduate school. But these thoughts of I can't or I am not smart enough still continue. But with the help of therapy and the unwavering support of my family, friends, teachers, and professors, I continue to show up and really try my best. Personally, I also like to reflect on the struggles I've overcome and the achievements I've made in the past. And I think to myself, well, I've made it this far. How much farther can I get? So now that I've shared with you where I have been and how I got here, I'm going to share what I am currently working on. So during my first year of the MCB program at Brandeis, I went through lab rotations and I also had some classes. The rotations are meant to allow students to try out a few different research fields and labs to find one they feel they'll fit best in. At the end of my first year, I chose the Cadner Lab, which studies RNA regulation. Just to make sure we're all on the same page, here is a diagram of the flow of genetic information. We start from DNA, which gets transcribed to RNA, and then is translated into protein. As I mentioned, we are interested in RNA regulation, so I'm going to dive a bit deeper into transcription. Starting with what a gene is. A gene is a distinct sequence of DNA that codes for functional RNA or protein, and it's made up of introns and exons. Here I have a diagram of what that looks like. Normally, once the RNA is transcribed, it undergoes RNA processing, during which the RNA is spliced. The exons of a gene in purple are needed to make the protein, so the introns in orange are removed. So we end up with an RNA that is made up of exons. This mRNA can then be translated into protein. Occasionally during this RNA processing, an exon is spliced out. and it ends up being circularized. This circular piece of RNA is known as a circRNA. Now the cell ends up with a linear RNA that has a different sequence than the one on the left. But this isn't always a bad thing. This is actually known as alternative splicing. These circRNAs are what my studies focus on. So let me tell you a little bit more about them. The presence of these circRNAs, interestingly, are most abundant in the brain compared to other tissues. They also increase with age. They're very stable because exonucleases are not able to cut them, and they are conserved, meaning they are found in mice, humans, and in my model organism, the fruit fly or Drosophila melanogaster. This is what we know about them, and many questions remain. One question is, what is the function of these circRNAs? They are abundant and they are found in all eukaryotes, so why are they there? We can use the fruit fly as a model organism to study these circRNAs because, like I mentioned, they have circRNAs in their brain tissues. They are also easier to handle and easier to age in comparison to mice. Since they've been studied for quite some time, we have lots of genetic information and tools that we can use to answer our questions. There are multiple ways that I can dig into this question of the function of circRNAs. One way being that we decrease the amount of a specific circRNA and see what happens. This idea of knocking down is very common in science. In a sense, it's like Jenga. If you take out a block, what will happen? Will the tower fall? Will it still stand just fine? Will it wobble? So if we decrease levels of a specific circRNA, we can look for a few things. Will the fly survive? Does it develop normally? How is its behavior, and daily activity levels? Are there other genes increasing or decreasing in a specific pathway? Another way that we can understand the function of circRNAs is by probing what we know about RNA regulation. AGO1 or Argonaut1 is a protein known to be involved in RNA regulation. It's bound to another RNA called a microRNA or miRNA. Once bound, and with the help of other proteins, it forms a large complex. Normally, this complex then goes around looking for mRNAs that contain a complementary sequence to the microRNA. Once it finds one, it starts the degradation process for the mRNA. So, seeing that we have this protein that interacts with other RNAs, my question is, do AGO1 and circRNAs interact? The Cadner lab actually recently found a few circRNAs that do interact with AGO1, and this is so cool. But it also leads to so many more questions. Like, so why are they interacting? Do they do something together? Also, how exactly are they interacting? These are my questions that I am working on answering for my thesis. Before I end, I want to leave you all with 3 takeaways. 1, you can also get paid to do research. I am not paying for my PhD in biology. I get a stipend, meaning I am getting paid, kind of like a job. Many PhD science degrees are this way because it takes a lot of time and effort to do research. So PhD programs want you to be able to focus all your time and effort on research. It's And number 2, it's so important to build a support group. This looks different person to person, and that's okay. I go to my family, friends, and professors for encouragement and support as I need, especially in those times of self-doubt. Lastly, science isn't finished. There are always new discoveries, and there is always something new to learn. Thank you all so much for listening, and I hope you enjoyed my presentation.